say Krebs?
Hey guys!
Hey
guys! This is, what is it?
The Krebs Cycle. Haha, because there's a crab.
Okay. It's actually the Krebs Cycle, also known as the Citric Acid Cycle. So, what is it? The Krebs Cycle is the second stage of aerobic respiration. It happens after glycolysis and before ETS. It in acetyl-CoA formation. It ends up breaking down pyruvate into carbon dioxide and 10 coenzymes, as you can see down by the products. And these coenzymes will eventually be used to deliver electrons to the ETS. So it's very important. And you can see here we've got the reactants, ADP, NAD+, and FADA. And it yields CO2, 2 ATP, and 10 coenzymes, as I said before.
The Krebs cycle was first introduced by Hans Krebs, who was originally an ear, nose, and throat surgeon. He discovered the Krebs cycle in 1937, and he then received a Nobel Prize for it in 1953.
So what's the point? The point of the Krebs cycle, as you can see in this photo, is to break down 2 pyruvate, which we get from glycolysis, into 6 CO2, which ends up yielding 2 ATP and 10 reduced coenzymes. So these coenzymes are extremely important to carry electrons and hydrogen ions to be used in the ETS, because without these coenzymes, it would be really hard to create that H+ gradient for the process of glucose to be made. So you can see that really well in this image.
Okay, the Krebs cycle occurs in the mitochondria matrix, which is the space within the inner membrane in eukaryotes. And then in prokaryotes, the Krebs cycle occurs in the cytoplasm because they don't have mitochondria.
So this is a diagram of what the Krebs cycle actually looks like. So the Krebs cycle starts here when pyruvic acid comes in from the first pre-steps to the citric acid cycle. It starts with glycolysis and fermentation, but that's another video. So the cycle flows like this, and the steps will be specified now.
So the steps begin, as Hannah mentioned, with the pre-steps, which 2 pyruvate enter the mitochondria, and the enzyme split the pyruvate into 2 carbon acetyl group, in which CO2 is released, as you can see, and it also creates the coenzyme acetyl-CoA, which NAD+ combines with hydrogen ions to form NADH. So then we go through this process of where NAD combines with hydrogen ions, and it converts to NADH. So this happens twice, so you can see right there. And then down here we have, this is phosphorylation. So ATP is being created. You can't see it because of this freaking thing. Oops. Sorry. But yeah, phosphorylation occurs right there. There's ATP being made. So then FAD combines with electrons to create FADH2. So at that point, you've got a bunch of coenzymes being made here, 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 and then eventually it comes back into oxaloacetate, don't know how to say it, but yeah.
Okay, so I have 2 cool fun facts for you guys. So one is that the synthesis of other biomolecules often also can occur within the intermediates of the cycle, and that the Krebs cycle is considered to be amphibolic because of its dual metabolic role that involves both catabolism and anabolism in its biochemical pathway.
So, how does this relate to photosynthesis? So, cellular respiration is basically just the inverse of photosynthesis. But the process in photosynthesis that is most similar to the Krebs cycle would definitely be the Calvin Benson cycle, because they're both cyclic and they are the closest to each other. However, the Krebs cycle is an aerobic process, unlike the Calvin cycle, which is considered anaerobic.
So the main takeaways that you should have gotten from this video is that you now know that the Krebs cycle is the second stage of aerobic respiration, which occurs in the inner matrix of the mitochondria in eukaryotes. And the most important thing is that we take pyruvate from glycolysis, turning that into acetyl-CoA and CO2, and most importantly, yielding 2 ATP and 10 coenzymes, which are going to be very important to carry on to the ETS.
That's all, folks.
Oh, crabs. Krabby Patties.
Make it stop.
Where's the thing?
Oh,
You
Where's
deleted
the
it.
pause button?
no.
Oh, no.
Did you delete it?
No.
Crabs.
Where'd it
Don't
go?
unmute it. No.
Lying.
I'm not doing it again.
You're lying.
Where'd it go? Wait, hold on. Maybe it's
lying.
my recordings. What?
I'm eliminating myself immediately.
Okay, well, I guess
I think when we click out of that box in the corner.
I won't do that this time.
Oh, wait. So what am I doing?
You're doing fun
Well, at
facts.
least I don't have to be cringy again.
Yeah, mine was pretty bad. It's fine.
I had no idea what to say.
Oh, wait. Are we still recording right now?
I
No.
don't think so.
Just kidding. I don't think we are. I don't
What
know,
if we are?
There's a red dot right there. And just... Oh, yeah, it is.